Muy buenas tardes a todos. Good afternoon, everyone. Good morning, Dani. Recuerden que pueden colocar la interpretación de esta presentación en español, ya que va a ser realizada originalmente en inglés. La pueden realizar apretando el mundito que sale en la parte inferior, casi derecha de la pantalla, donde sale interpretación. ¿Sí? Bien. So, I will start introducing Dani. Dani is the principal investigator of our project, Plasmodium. So Dani has a master's degree in experimental physics from the University of Leiden in the Netherlands and has a PhD in applied physics from the University of Linköping in Sweden. He's mainly interested in multidisciplinary projects related to physics, engineering, and biology, especially the application of micro and nanotechnology for life science issues. Particularly, he's interested in organ-on-a-chip. He has held positions in the best universities of the world, such as Princeton University, Seoul National University, the National University of Singapore, working on research topics such as biosensors, DNA computation in microfluidics, 3D cell culture in microfluidics, human on a chip, organ on a chip, microfabricated structures, automated liquid management. He has published more than 40 articles in international journals. And also he has 4 patents. So welcome, Dani, and you can start the presentation of the project.
Okay, good morning, Rocio. Thank you for your introduction. I will share my screen now.
Sure.
So I hope you can all see the screen.
It can be seen.
Okay, so I'll talk slowly because I realize there's an interpreter who has to keep up with me. So good evening, everybody. For me, it's morning. I am at the moment in Singapore, giving you this presentation. So this presentation is actually about microfluidics and what we can do with that for the diagnostics of malaria. So the question is, what is malaria? Why malaria? I think most of you probably know that. There are 228 million cases of malaria worldwide a few years ago. And so that influences a lot of people, mainly in developing countries, South America, and also actually more severe in Africa. There are also cases up in Asia, in the Southeast Asia. Basically everywhere where it's tropical, you will have this. So it doesn't, but that doesn't only affect people from a disease point of view, but also it has some negative effect on the gross domestic product with countries who have very high infection rates. So this is the parasite, the malaria parasites. There are a few different types, but they all are, you get infected by the mosquito who bites you. And then we have this sporocytes, which is called, let's see if this mouse works. Sporocytes, which get into the liver, and from the liver, it gets infected the liver cells, which is called hepatocytes, and they burst open, and then they infect the blood cells. And once we're in the blood cell stage, the blood cells are infected, the parasite grows, and then it bursts again, and then we have the whole cycle repeating. So this is the stage which is important. This is the stage we have to prevent, actually. But if we can't prevent it, we have to find it in patients. And of course, that is the problem. You want to detect it as soon as possible. So that means you want to be able to detect low quantities of the infected blood cells. So just to give you some numbers, for Peru in the last, well, from 2015, 2020, we have the numbers. And you see, the numbers are quite high, but that they are dropping. But that can be for several reasons they are dropping. So this is a worldwide trend, but it doesn't mean that the disease is cured. Actually, it is still a big problem, malaria. Although there are talks of cures recently, it does not mean it works. And as you might know, except for COVID, usually drug development or prevention development takes many years, at least 10 years to do that. For COVID was a different story. It just everybody was on board, and it took a very short time to develop the drugs. But you must realize that a lot of groundwork was already laid down to do that. So, what is the problem with malaria? What are the challenges? So the problem is you have to detect the malaria as soon as possible. If you're too late, it's too late. You cannot do anything anymore. So you have to find malaria very timely. So you need diagnostics which can do that. It's also in developing countries, you have, of course, the difficulty of finding specialized health centers and trained personnel. I mean, usually malaria is in the poorer parts of the country with little access. So actually you would need some system which anybody could use in any location. Also, there's you might think there's a lot of parasites in the blood, but there are not so many parasites. So you need a very good detection limit to actually find them. But the more so, if you want to find them earlier in early stages, you need a high detection limit to do that. Then there's also that you get from malaria, you get acute fever, but there are also other endemic diseases which have that. So you're not, even if you have the symptoms, it might not be malaria. It could be other things. And then there is the problem, which you might have heard also in the news, is drug resistance. Since we're taking a lot of drugs in the world, viruses, bacteria, and parasites are getting drug resistance. They actually evolved to thrive in drugs. There are some experiments on that, which was done by a friend of mine in Princeton University. He had E. coli, and he showed that actually E. coli could survive in a four times deadly dose of drugs and thrive there. Although everybody thought they should be killed, but they weren't. They can mutate, or he says evolve, and get drug resistance. So the more drugs we take, the more resistance there will be. And there are other that the organism itself will get resistance. So in this case, that the mosquito will get resistance of the pesticides being used to kill them. But that doesn't always work either. I mean, the more we spray them with chemicals, the more they will adapt to it. So what we need is a cheap, simple, and reliable diagnostic system which can be used at remote locations. And there are some keywords here, which is cheap, simple, reliable, and remote. So this is something you have to work on, or which is the aim of the project, was to work on. So now a bit of an introduction about the project we've been doing over the last 2 years here. So it is aimed to develop a low-cost method of concentrating of pre-concentrating and identifying malaria infection in a blood sample. So the pre-concentration is done using microfluidic separation by leveraging mechanical changes caused by the infection. So actually, the infected blood cell has different properties than healthy blood cells. And then once they are separated or concentrated, because when you concentrate it, it's much easier to do the diagnostics. That's one of the reasons to do the concentration. So, and then we do the diagnostics, the identification by bioelectric changes caused by the red blood cells. Because of the different properties, the red blood cells will give a different bioelectrical signal. So as I said, the changes in the properties of the red blood cells is usually in the cell morphology, so the shape of the cell. It increased rigidity, they become stiffer. They have an elevated permeability and changes in iron content. The change in the iron content is, of course, one of the reasons why you will get the problems with malaria, because changing iron content, the iron content is getting lower, so the red blood cell takes up less oxygen. And of course, if the iron content becomes lower, the electrical signal, so actually the electric resistance of the cell will also become higher. And therefore we can see the changes. So the diseases you can get is cerebral malaria or anemia, and can be used to separate and identify infected cells from healthy ones. So the system we are designing and trying to build is actually 3 steps. We have the initial, so we have to extract the blood from the patients. So one thing about malaria is that you cannot culture it, so we have to extract the blood from patients. And then we can do the first separation by inertia, the size, because we have also the size between healthy and infected blood cells is different. So we can separate it on size, and then we can also separate them on deformability. That is to clean up a little bit more. Deformability means that because the healthy cells are softer than the infected ones. And once we've done a good separation of the healthy and infected cells, we can do the bioelectric measurements, that is called with impedance analysis. Each module, so here are some technical problems, or each module will have different flow velocities. For size separations, we need an 18 microliter per second flow velocity. For deformability, we need a 40 microliter per second flow velocity. And when we do the analysis, we have to go down to 1 microliter per second velocity. So somehow we have to engineer the system in such a way that the flow velocities in every module is as indicated here. So it's just an overview of the system setup. So we have the inertial module, which is a spiral. I'll show you later how that sort of works. We inject this module with blood from a syringe pump. It gets pumped in, and then we separate out the healthy blood cells. Oops. And we separate the infected blood cells, in this case, which we're interested in with Plasmodium. Once we have the infected, that goes into the other module. And then the impedance module, we will try to align the cells in what we call a train of cells. We need the cells to be in one line, because they go through a set of microelectrodes. They have to flow over it, and we want to see them one by one going over the electrodes. So the cell will be between the electrodes, and then we will measure with the impedance analyzer, we will measure the properties, bioelectric properties, and we can record it on the computer, and then we get something, what you see here in this graph. We will have a peaks, and then we know a red blood cell has passed by. So there will be a difference in signal between the healthy blood cells and the infected blood cells. Because even if we do the separation, there will always be some healthy blood cells in there. You're never going to have 100% separation. So we go to the first part, that is the particle or separation by inertial flow. So we tested everything with particles, with microbeads, because it's we shouldn't start with blood immediately. So the idea is we have a spiral, and in a spiral we put in a mixed solution with different kinds, sizes of beads. They go around and they will start to separate. They'll line up at different sizes in distinctive bands. And then in the end, we can separate them out through different outputs. So, of course, this is the ideal case, that everything's perfectly separated out. It's not going to work. You see two inputs here. One is for sheath flow. There are two ways to operate this. Is one just to put directly all the beads in and have them separate out, or you can add an extra element in here, which is sheath flow, which pushes all the particles to one side, and then they have to separate it out. So it has sometimes advantages and sometimes not. Different methods for the same things. So because of the spiral, there are some different forces playing on the particles. So one is the lift force, which is a function. So the flows are laminar. That means laminar is opposed to what you see when you open a tap, that is chaotic flow. So laminar is a very well-defined flow, and it has a parabolic velocity profile. And it only appears in very small channels. It's called the Reynolds number, and that depends on the channel size, the viscosity of the flow, the fluid, the flow velocity, and things like that. I won't go in details there. So we have a parabolic fluid velocity profile, which pushes towards the wall. As you can see, there's at the edge of the flow, it will push this particle towards the wall. And but since the particle goes to the wall, there's also an interaction with the wall, which will push it back to the center. So we have an interplay of forces of where the particle goes to, and it depends on the size of the particle, how far it will stabilize itself, that these forces are in equilibrium in the flow. So it will be in different distances from the wall, depending on the particle size. There's also another force in effect, this is the Dean's force, which is a function of the vortices created by the fluid flowing in a curvilinear channel, which means the spiral. That's why it's a spiral, to create a curve and create this Dean force. So there's an extra force, and depending on the size of the particle, again, it will be bigger or smaller and separate out. And if you have deformable particles, there is an extra lift force from the deformability, which is in the same direction as Dean's force. So this is all a bit technical, but it's just to show you that there are forces at play in this spiral, and you have a little bit of sense that they are able to separate out different sizes of particles. So that's a bit of the theory behind it. We did simulations to see how the particles are separated, at which flow rates they separated well, how many turnings we need for the spiral, because it depends on the length of the spiral and how many windings the spiral has. It's not that the longer the spiral, the better the separation, because it doesn't work like that. So that has all been done. So we came up with the parameters, the sizes, and the number of windings for the spiral, and then we have to make it. Since this is virtual, I cannot say Louis here has made it. Louis is somewhere on the Zoom call. So in an overview, we actually 3D print a mold, which is UV cured. Then we have something which is called PDMS, it's polydimethyl silane. It's like a silicone rubber. It's a 2-component which you mix together. It has a curing agent which you mix together, and then you degas it, because when you mix things together you have bubbles in it, especially when it's very viscous, so you have to get the bubbles out. And then you pour it in to the mold, and you cure it by heating it up for 80 degrees for 20-30 minutes. And once it's cured, you peel it off, you stick it to a piece of glass, but the glass has to be, and this PDMS, what we call a PDMS chip, has to be cleaned with the plasma bonding. It's an oxygen plasma bonding. It gives you active groups on the surface, and then we can stick them together, and it's permanently bonded, so it's stuck. And then you do the testing. So we go through the steps sort of a little bit closer. So we 3D print the mold. So there's a 3D printing, stereolithography 3D printer. That is a device which has, as you see, well, the devices on the one we use is on the left side, and on the right side is sort of the principle. There is a tray with what they call the photopolymer. This is a resin which, with UV light, starts curing. This is in an XYZ stage. Oh, sorry, this is just in the Z stage. It moves up and down. So we actually pull out the object we're printing from the liquid. And to do the XY, there is a scanning galvanometer. That's a mirror which is moving for the XY direction. So we can expose different areas in this in the bath of resin. And this is controlled, exposed with a UV laser. So it's the UV cures or hardens the resist. It's just like taking pictures, except with a bit different chemicals. So this we have standing in our lab. The only problem with this is that the resolution is not as good as we hoped for. So we had to optimize the whole printing process, which I'm not going to show you here. But you make a drawing in the program, and when you print it, the sizes are a little bit different. So you have to go back to your drawing, change the sizes to make sure you get the sizes you really want. So it's not all straightforward at this scale. Since we're in the micrometer scales, the channel width is 200 micrometer. Channel width height is also 50 to 100 micrometers. So these are the scales we're talking about. Just to give an example how this looks like, you can see the stage here, and this structure gets pulled out of this reservoir that you see here. with on the bottom with the resin, it gets pulled out, and that's how you get your structure. Ours doesn't really look like this, but it's just an example. So here's the close-up of the mold Louis made. It's you can see the spiral here. It has the input in the middle, and then it has the output here at the side. So this is made with this 3D printer. So 3D printers, why do we use the 3D printers? For one thing, it's the only thing we have in the lab. And for the other thing, it's a cheap way of doing prototyping. Usually you will make chips like this in clean rooms with what they call the lithography, lithography. And then you can make much smaller features than with 3D printing. The problem is there are no such facilities in Peru. The closest clean room is in Bogota, and there are a few more in South America, but not many. So usually clean rooms are used to make electronics chips, but we also use them to make microfluidic chips, especially if you want very good ones and very high resolutions. Since we don't have them, that's why we go to the 3D printers. So these are used with the Formlabs 2. That's the brand of the 3D printer, and we use a clear commercial resin. You can see it's opaque. The reason why it's not completely clear is because the surface is a little bit rough. Because with the printing, you will never get a very extremely smooth surface. So then we have the PDMS. This is sort of a rubbery silicone solution, and which we have. You can see in this picture here the base liquid, and we have the cure. We mix them together 1 to 10, mix them up. So you need a weighing scale to mix them. You mix them up, and then you pour them in the mold, as we see here. Then we throw it in a vacuum. So to get the degassing, we get all the bubbles out, because we do not want bubbles in our chip, because if we have bubbles, then we cannot see things very well, for one thing, and the bubbles are bigger than the channels itself. So that would give a lot of problems. Once we degassed and cured it, we clean the glass slide. We're going to put it on the glass slide. We clean it with the PDMS chip as well in the oxygen plasma to make those active groups on the surfaces. Then we put them together, and they are, as we said, bonded to the glass irreversibly. You cannot peel it off anymore, which is good because we're going to do the flow and it's going to be high pressure. So here's the experimental setup. Here you see the Louise's hand. There's the pump. We're using the chip you can see on the microscope slide here with the channels, and on the monitor you can see with the camera what is happening in your chip. So for the experiment, to see if this all really works, we use the particles from 10 and 6 micrometer in diameter. Just to give you an idea that that's about the same range as blood cells, red blood cells are. So we are in the right range, but not with the right material. So we first try the polystyrene particles to see if we can separate. These are particles are suspended in water and have a surfactant, and then we pump them, flow them into the chip at different speeds to test which speed is the best. We did the simulations, but still in an experimental phase, we still want to see if it's correct, because simulations are not always correct. So we get results by counting the particles to see at which output are which particles. So this is an image of the of our experiment at work. We can see the small dots, red dots, which are the small particles flowing, and we have the green ones, which are the larger particles. Sometimes they get stuck to the chip. That's why you can see them, these dots here. But you can also see the flow. And as you can observe from the inner outlets, most of this green is going through that channel. There is no, or only a few particles, green particles going through the outer outlets. This is not completely true for the red particles. So we see more red particles going into the inner outlets. But most of them are still going through the outer outlets. So you can see a clear separation. You can see these lines, the red line flowing here. So that's the particles in motion, and the green line is the green particles in motion. So you see a clear separation of the particles by using this simple device. So we did experiments to see which is the best flow velocity to get the optimal output. And we found that the flow of 16.6 microliters per second, we obtained a 98% separation of the big particles, the 10 micrometer particles, those were the green ones, and a 67% separation of the 6 micrometer particles. This is actually quite good for the system we built, because getting a 98% separation is not easy. And although you might think this the smaller particles are not so well separated, you have to think that if we wanted to have the 10 micrometer particles, it is almost pure. So if this is our focus group, we've done a very good job to separate them, and we can use them for the next step. So if we're going to lose some material on the way, so that's okay, as long as we have the particles we want. So we can see at different flow velocities that we have the 98% separation at the 16.6. So you can see that at 15.2, although the separation looks slightly higher, the difference in separation is not so big. So we need it in the inner outlet. So the 16.6 microliter gives you the best separation. So this is the speed which is optimal for this experiment. It has to be said, if you change your particle size, you will also probably have to change your flow rates. If you change the channel size and the windings, you have to change the flow rates. So you really have to optimize the system to be able to do this. So then we go to diagnostics, because we want to see if we really have the malaria affected cells. So here's an image I found, and you can see the difference between the malaria affected cells, the one cell in the middle here, red blood cell, with the non-infected cell. So you can clearly, or we can clearly see the difference if we have a very strong microscope, high magnifications to look at it. But of course, you cannot screen patients' blood like this, because it will take you ages to count all the cells. So one of the most used methods these days to do diagnostics is what they call blood smear. So they put a bit of drop on a slide, and then they smear it over the slide to make a thin layer of blood. And then you can put it under the microscope. And then you will see something like this. You see the individual red blood cells, and once in a while you see a cell which has a parasite in them. Of course, you can see there's only one or two cells here which are infected on this whole few. That means if you want to know how serious it is, you have to do a lot of these samples and try to screen them all. And of course, you might have the bad luck. There is no, or the good luck, it depends how you look at it, that there is no infected cell in your image. It doesn't mean you do not have malaria. It just means you didn't see it in that one image. So you have to go through a whole different set of images to find this. So this is all done manually. So there's no automation or what. So it works, but it's not foolproof, and it's certainly not foolproof. And it's very laborious to do this. So that's why we wanted to come up with a different way. And one way is to do the impedance measurements. So in impedance measurements, that means we have electrodes parallel, and when there is a blood cell in the middle, we can measure the properties, the impedance properties of this cell. So that means the resistance basically of the cell, because we put a electric field over here, and then we measure the resistance. So that means in the channel we have this chip, the PDMS channel, in which we have the electrodes, and then the blood cells come in one by one, and then we can measure them. So we would do this for beads. This is just from an article. You could see, people have measured that you can see the difference between plain beads and functionalized beads with a protein on it, a carboxyl bead. Sorry, not protein on it, just a carboxyl group on the bead. And you can see already a difference in impedance. So it's a quite sensitive method. And of course, being very sensitive, it also means it's very sensitive to disturbances. So you have to take care of that one. So just to give you an idea how big or small this is. So this we make this on actually a collaborator of us in China made this on a standard microscope slide, which is 75 times 50 millimeter slides. We put on the electrodes by gold deposition. It's either a titanium layer first and then the gold, so there's the E-beam preparation, or you put a chrome layer first and then the gold on top. You need these adhesive layers first, because gold does not stick to glass, it will just fall off. So the width of these electrodes is only 20 micrometers. The length is 20 millimeters. The gap size between the electrodes is also 20 micrometers, while our channel width in this case is, the overlap of the electrodes is 340 micrometers. Just to give you an idea how much 20 micrometers is, that is about 1/7 of the width of your hair. Just gives you a rough idea. So we have this in the channel. This is a cross section of the channel. This is you can see the glass at the bottom, the gray, and the PDMS on top. And then we have the electrodes in the channel here. The channel is 200 micrometer wide and 30 micrometer high. There is a little bit layer of PDMS still over the electrodes, because the electrodes cannot be exposed directly to the water, because else we just short circuit it. And then the cell will be on top, and then we can measure the impedance. So going back to the slide, what you see here, the cell has to be on top of the microelectrodes. We just want one cell there, and we want this cell close to the electrodes, because the closer you are to the electrodes, the better the measurement. So we have to push the cells in the center of this channel, and we have to push the cells down. So we can do this with the hydrodynamic focusing. So we have here's just a little bit of overview. We have the sample coming in, and we have to push the cells down to the bottom towards the glass and the electrodes. And well, this one is the three-dimensional focusing, but we also have to do the same focusings from the side to make sure that these particles go, or the cells go into the center of the electrodes. We want to create a train or a line of cells or particles. So to do this, we have a chip here with a lot of different inputs. We have, so this on the left side, you see the mold and the channel, and on the right side, it has the indicators of what is what. We have first the 2D focusing, and as you see on the left side, it splits in two and then pushes. And on the second pillar, when it comes together, these channels, it will push that flow to the center. And then we have the sample input, which comes in, then it's pushed to the center by the 2D focusing. And then we have a 3D focusing, which pushes the sample down towards the bottom. And then we have the outlet. So that means you need 3 pumps to run this. Here is just a sort of the real chip you can see here. The electrodes are connected to the wires with the silver glue, and then you have the inputs and outputs here in the blue. And you can see the size. There's a screw here, so you can see about what the sizes of this chip is. As I said, it becomes a little bit more complicated. There are now 3 pumps involved to get the flow in there, still on top of the microscope. The chip is now mounted sideways to have a better look at the focusing, especially the 3D focusing. So here we see the results of the 2D hydrodynamic focusing. So depending on the flow rates that come from the sides, you will see a thinner and thinner line. So that means we can focus the beam. These are done on fluorescence beads, so they light up, else you cannot see what's happening at this speed. And the little dots you see here are beads stuck at the center, at the edge of the chip. They sometimes stick there. We can also do this in the 3D focusing. So different flow rates, we can push down. This is so the side view of the flow. We can push down the flows, and you can see if we at a higher flow rate, we can push it down the flows of the particles. The arrow indicates the edge of the channel. So you can see in figure 1, it's a complete the particles all over the place, while in figure 4 we managed to make the particle stream only about 15 micrometers high. And keep in mind that the particle is about 10 micrometers. So that's quite good. And since we're doing impedance measurements, which are very sensitive to, as we found out, to simple things like the lights in your lab, because light tubes or electricity has a 50 hertz, also 60 hertz. The current has 60 hertz, so you can see that when you have this chip. So we have to shield it. So the chip is now shielded with this copper cage. So we keep out all the signals we do not want. Also, if you bump the table, you will also see the signal. So it has to be isolated. This is just an image of you can see the electrodes here, and you can see the beads coming in, and one bead is here between the electrodes. So this is basically what we're looking for. And then we can do the impedance measurements. These are the sort of the first results we have on particles. There is still some noise, there always will be noise, but we can see the peaks of the particles going over the electrodes. So our next stage will be to do the malaria cells. Coming back to the malaria, because so we still have to do experiments on the malaria, but due to COVID, we had some problems getting the malaria cells. So as I said, we cannot culture, in this case, Plasmodium sample we wanted. We cannot culture them, so we cannot culture them in the blood. So we need patient's blood. But because of the COVID, the attention was not on malaria at the moment. It was on COVID and how to get everybody vaccinated and treated. So all the clinics were more busy with that. And also due to the elections, there were changes in government departments, especially the health departments, and from which we would have to get the malaria samples. Weren't responding very well. So finally, a few weeks ago, we got some frozen Plasmodium samples, which is not what we wanted. But we can still use them for another test, but we can't use them to do the separation. We cannot infect red blood cells with it. So we still don't have those samples. Still working on that. But we can do some tests because you might have malaria. We can show you, we can diagnose you with malaria with the system I just showed you. But you could also still do another test, sort of a simple test as well, although it needs more reagents, so it gets more expensive. It gets a bit harder to do. With which we can determine which malaria you have, because there's a few different types. Plasmodium being the most important one, at least in Peru. So we wanted to see which one you have, and you can do this with what they call sandwich ELISA. And the sandwich ELISA is so these are wells from a 96 well plate, and you put in a capture antibody, and then you do your test with your sample, which has an antigen which binds to the antibody, and then you're going to do detection with a similar antibody, which has a label, which is a biotin label, which attaches to your antigen, and that label you're going to use a strep with a HRPO, which at the moment I do not know what that is, a fluorophore. So it will light up when you put another substrate through it, that you will have a colored product. So you can see the colorimetry. You can see that there you have the antigen, so the infection. If the antigen is not there, the secondary antibody in number 3 will not bind, so this whole process will not work. So Rocio, who's also in this talk, has done some measurements on recombinant material and monoclonal antibody just to test the detection limits of negative controls, positive controls for the Eliza system. And you can only go to 0.02 concentration for detection limit. In the samples you tried, the Mustra was at the edge of the detection limit. So there's still some work need to be done to get the detection under control. This is at the absorption of 450 nanometer light to do the test. Negative controls, they all work pretty well, and the positive controls with different concentrations will give you different, so a different concentration will give you different detections. But since the material is very low in concentration, it is very hard to do the detection. Since we have the frozen Plasmodium, Rocio started with some preliminary measurements on the Eliza with different concentrations of the Plasmodium. As you can see at the x-axis here, and with different antibodies, primary antibodies for the Eliza concentration, of course. And then she did this preliminary test, and in principle, the more Plasmodium you have, the higher your detection will be. Unfortunately, it's a little bit, the signal's a little bit noisy. This is the first measurement, so there will be more measurements done, and then we can put in the error bars and see how significant the results are. So actually what you want, so this is not, hasn't been made, this a long time ago, somebody else. What you want to do is, like we did our COVID testing, you also want to do like a malaria test on a rapid diagnostics test. It's just a piece of paper with your antibodies on it. And then you can detect from your blood which type of malaria you have, or if you don't have them. Now, so going to the future, and so what can we actually sort of do with this? So, as I said, what we actually want is a simple, reproducible diagnosis of patient's blood. That's one thing. We want to do diagnosis, but the other thing we can do is actually filter out the red blood cells from the patients. As you see, we can do with the spiral, we can do the filtering. So we can separate out the infected and the non-infected red blood cells. So this is like the same principle as you see in the dialysis machine, a kidney dialysis, where you hook up a patient to a machine and clean the bodily fluids. In this case, we can do it also with the blood and clean out the blood. So to do the diagnostics, you saw what we have here, and on the left side, this is our setup. And actually, there's more to the setup than just what you see in this picture. And everything will have to fit in a small box, what you see on the right side. This is just to point out, not our box is just an example of what a small, simple diagnostic system should look like. So we have to pack everything we have here on the left side into this right box. That's the first thing we have to do. And to do the dialysis, the separation of the red blood cells from patients, we have this, as you saw before, we have the spiral, we have the mix coming in, and we separate it in the end, in the out. So this is a module for a spiral, and which you see in the center. And you saw it was quite small. So to handle, say, 5-liter blood, we would need, a few thousand of these in parallel. And I just put here, just think computer chips. There's a lot of small components on computer chips all put together, and you can handle something bigger. So we can do the same with the microfluidics. We just get a lot of these and put them together and so we can handle the blood. So we can have this module, we just stack them up, and once they're all stacked up, we can connect them to the cell's input, as I call a paralyzer module. We have the separation modules, and then we have the output with the different separated out, and then we can put them together and put them back into the patient again. Of course, this is the simple view. There's a few more steps which has to be done, because one thing, the blood is too thick to go through here, so we have to dilute the blood. And once we dilute the blood on the other side, we have to get the excess of liquid out of the blood to do this dialysis. And then in the end, we would like to have something like you see here, which is connected to the patient and just separated the malaria infected blood. You can also, the same principle can also be used for sickle cells, also a different kind of blood disease, or even had in mind for sepsis, that means bacteria in your blood, which you would like to get out. So that's also a possibility to do with this kind of spirals in microfluidics. So this is the end of my presentation. So I'd like to acknowledge all these people who were involved. Was Nicolas Thorne, he did all the simulations mainly, and other tasks as well. Louis Nestor Flores, who's at this talk, he done the fabrication and the experiments, all the mechanical ones. Rocio Egarville, she has done the biological work. And then we have Julian Noel, Emir Augusto Vela, and Julio at the end, which are the professors at UTEC in this department from 3 different groups. Romero Moro, who is not in China anymore, but was in China. He helped us fabricating the gold electrodes on the glass slides, because that's something we cannot do in Peru. Aaron Fleischman, he's a scientist in Cleveland Clinic in USA, who is also has a lot of experience in these things and has advised us a lot with this project. And then I leave you with this image of UTEC, the staircases, which reminds me a lot of this image from Escher. So you sort of, when I walk around UTEC, I get a bit confused as well as which staircase I have to take. So that concludes my presentation. So you can ask questions now. Louis and Rocio are there as well, and you can ask the questions to them in Spanish if you want. So I thank you for your attention, and hope you learned a little bit about this talk and could follow some of it. Thank you.
Thank you very much, Danny. I would like to especially thank Naval Medical Research Unit 6, Namru 6, for providing us the latest Plasmodium samples. All right, so I would like to know if we have any questions from the public. You can ask in Spanish or English if you want to. Okay, so we have some questions. Henry Ventura is asking, what type of detection are we applying?
What type of detection? Meaning which? Well, it's a bit of a strange question. What type of detection are we applying? So we did 2 types of detection. One was with the impedance detection. So there's just measuring the resistance of the cell. There's one detection. And the other one was the Eliza. There's another form of detection. For the system, we want to use the impedance. The Eliza is like a checkup to see if we do everything correct, but it could also be used independently, but it is more expensive to operate because we need all the reagents, which are sometimes not very easy to get, I understood. So that's why we wanted to go to the impedance measurements. So that's the type we are focusing on. Hope that answered your question, Henry.
All right, thank you. Harry Saavedra is raising his hand. So, Harry, please.
So Harry Saavedra wants to know about the electrodes.
Yeah, electrodes,
Why
titanium
the
and
titanium and chromium, as I said in the talk, these metals are used as adhesion layer for gold. Gold does not stick to glass. So we need another metal in between to bond, to sort of glue the gold to the glass. And that's why we use either titanium or chromium first. So that this is evaporated to the glass first before we evaporate the gold there. So that's the only reason why we use that. There's no other reason to use titanium or chromium except for as an adhesion layer.
Okay, here there is another question. With the intention of creating a device that filters infected blood cells from the patient, could combining the current technique with magnetophoresis enhance separation sensitivity?
Yes, actually, the person I should acknowledge at the end, Aaron Fleischman at the Cleveland clinics, has been looking at this way of separation by using the magnetic property of the red blood cells, because there is difference in the magnetic property of infected red blood cells and healthy red blood cells. So it can be used. We have opted the other way with the spiral, because the spiral is what is much lower cost than doing the magnetophoresis to make that. But we could, as you pointed out, we could combine both and to enhance the separation even more. We have been thinking about that, but we have to sort of focus on one thing at a time and not do everything at the same time, because then we sort of get lost. So, but it's something that has been on our minds and still in the back of our minds, if we should add that magnetic phoresis to do it.
Okay, thank you. I think Harry has another question.
And Harry said thanks.
Okay. Harry, if you have any other question, you can talk.
Okay, not really, but I just want to say thank you.
All right.
Oh,
yes.
Thank you as well, Harry.
Yeah. Yes. Yes, I saw you that you're walking around the UTEC around 1-2 months ago. But my other question is when you're coming back.
That's a good question. I do not know that answer. Actually, the project has been finished, so it all depends what the people at UTEC would want to do as next step. So let's see if we come back. There might be other collaborations coming up or other projects. I'm also introducing the department with people from Korea who are eager to come or to collaborate. So there can be some exciting times coming ahead. So we still have to see.
Okay, thank you.
Also, if I come next time, you can go for a coffee or tea.
Okay, there's another question. According to the question, I think related to combining the current technique with magnetophoresis enhance separation sensitivity. Yoyan is asking, a possibility could be parallel vertical design to include magnetic fields.
Yes, so then we're back to the magnetophoresis again. As I said, yeah, we can do that. We can include the magnetic fields, but it's a step we didn't want to take yet. So yeah, we can design it. It has been done already, as I said, by Aaron Fleischman in Cleveland Clinic. He has done separations with the magnetic fields. So this is a known technique already, which we could implement, but which we have not yet done, as I said, because we are focusing on the impedance measurements.
All right, and here we have another question. Hi, regarding the spiral for particle separation, you characterized the spiral to be a three-way round spiral. Is there any other study which used this spiral, I guess, as well? And if you add or modify this spiral, the rate of the separation might improve in any way?
Yes, to answer the last part, we done simulations so that's why we've done simulations to find out what the optimal shape was of the spiral. That you can, depending on the number of windings, you can figure that out. How the spiral should look like to do a certain separation. So that means how many windings you have, the size, actually the distance between the windings as well, the height of the channel, what do we call that, the ratio between the width and the height of the channel is very important, the aspect ratio. So these are all aspects you can change to modify the spiral, and that depends clearly on the system on the particles you are separating or what you are separating. And the spiral actually has been first used to separate out CTCs, circulating tumor cells from blood. So there's actually in Singapore, there's a large research was going on to that, to separate out the cancer cells from blood, what they call CTCs. They also use a spiral. And I was just talking last Sunday, that was yesterday, to a friend of mine from Korea who was also making a spiral. And something I haven't mentioned is the spiral can be trapezoidal. So the left side is higher than the right side. So we have done that. So we have changed the sort of the dimensions of the spiral, the shape of the spiral channels. Only on one side. My friend in Korea has done it on two sides. Where's my hand? So we can it goes from high to low from both sides, and that even separates out more. So you can play around with the sort of the structure, the architectural design of the spiral to optimize. There are many factors involved actually. So that's first why we did the simulations, to find out what the best way is to make that spiral. And well, so like CTCs, the cancer cells and healthy cells, the only difference there is the deformability. So you can, even with the same size but different rigidities of the cells, you can separate out, but you have to fine tune the spiral for that. I hope that answers the question.
I think it did. He says, thank you. Yes. All right. Thank you very much. If If we don't have any other question, well, thank you, Danny. Thank you very much for your presentation. And I hope we can have you in UTEC again very soon.
Okay, thank you for inviting me for this presentation. And indeed, I hope I can come back to UTEC again.
All right.
And I wish you all a pleasant evening there. And I just started my day here, so yeah, I still have a whole day ahead.
Have a great day, Danny.
Okay, thank you all. Okay.
Muchas
Bye-bye.
gracias a todos por su asistencia. Hasta otra oportunidad.